So welcome to the Sudden Arrhythmia Death Syndromes Foundation's Living with SAD series. Tonight's topic is Gene Therapy Basics and is presented by Tanaya Therapeutics. I'm Marcia Baker, the Program Director for the SADS Foundation. It's my pleasure now to introduce our speaker, Dr. Matthew Pollman. Dr. Matt Pollman is the Senior Vice President of Clinical Development at Tanaya Therapeutics. He is a cardiologist and a molecular biologist by training and has served in senior roles at companies such as Navigate Cardiac Structures and CV Ingenuity, as well as Abbott Vascular prior to joining Tenaya. Dr. Pollman received his medical training at the University of California, San Francisco, Stanford University, and Brigham and Women's Hospital. He has served as a full-time faculty member at Harvard Medical School and Morehouse School of Medicine. Dr. Pollman is author of numerous peer-reviewed journal articles and the past recipient of 2 investigator-initiated NIH NHLBI R01 grants in basic cardiovascular research. He has served many roles in the cardiovascular professional society space and reviewer of several leading journals in the cardiovascular sciences. So thank you, Matt, for volunteering your time today. And on behalf of the SADS Foundation, we thank Tanaya Therapeutics for your support to make this webinar possible. We'll have time for questions and answers following the presentation, and you may post them in the chat. Now I'll turn this over to Dr. Matt Pollman.
Great, thank you so much, Marcia, for that introduction, and want to thank the SADS Foundation organizers for putting together this series and inviting us to participate. It's certainly a privilege and an honor, and I'm very much humbled for the opportunity to do so. I also want to thank the participants this evening and those that will be viewing this later. Certainly you have a lot of different ways you can spend your time, and so we appreciate you spending the time with us. So we hope to make this very productive. And as Marcia indicated, we will be having a Q&A session toward the end, so we very much look forward to that. I think just a few comments before I start the presentation. I was speaking with Marcia just prior and saying in all of my decades of interacting with patients with various forms of cardiovascular disease, I certainly find the PKP2 community as one of the most informed, interested and informed communities that I've come across. So thank you for that, and looking forward to that continuing this evening. So let's talk about what we hope to accomplish over the course of the next hour. First, I want to get you to know Tanaya Therapeutics as a company, what we're trying to do, what we're trying to accomplish, where we are in our development programs. Then to provide, I think, some basic vocabulary, some building blocks as it relates to genetics, so we can understand why gene therapy, how gene therapy, and talk through some of the basics. Next, we'll cover some of the goals, some of the risks, as well as some of the promising opportunities and benefits of gene therapy. And then finally, to drill down a little bit more into one of our development programs, our TN401, which is a gene therapy for ARVC, but specifically ARVC caused by mutations in the PKP2 gene. So let's start by introducing you to Tanaya. From the outset, about 6 years ago now, Tanaya decided that they weren't going to approach therapeutics in an incremental manner and to make just incremental changes in therapies, but to transform and extend the lives of people and families fighting heart disease. So we wanted to make sure that whatever we take on, it's big, it's audacious, it's transformative for the patients' lives who could be affected by our therapies. And we decided we're going to tackle this in all forms, in that we not only are working on discovering new approaches, but developing and also delivering potentially curative therapies that address the very underlying cause of heart disease. And we hope to provide really new options for millions of individuals and families that are affected not only by rare genotype genetic cardiomyopathies, but also the most prevalent forms of heart failure. And we'll introduce you to our pipeline in just a minute. But there's two things that we really want to have you take away about Tenaya. At the very heart of this, we are 100% focused on heart disease, and we're driven by patients. And that's why we're here this evening. That's why I wanted to have this opportunity to speak directly to you and to hear directly from you. If you look at the pillars upon which the company is built, our values, and you look at the bottom there, it's patients first. So what does that mean? Is that in everything that we do, we want to keep you, the patient, in mind. Because if at the end of what we do, the billions of dollars spent, the years spent in developing something, if you as a patient don't come running to the therapy, embrace the therapy, are transformed by the therapy, we will have failed. And we don't want to fail. So all along this process, we have been listening to you as the patient, to learn what it is that affects you, what you would like to see from a therapy, and then incorporate that in our research and development programs. So we have a strong foundation in research, not only what we do internally, but we're constantly looking out on the horizon and understanding what are the emerging technologies that may complement and supplement what we're doing. And then we have a development organization that takes these new technologies and drives them toward development and clinical readiness. And it takes really this integration of all of that in order to deliver on our promise to make transformative therapies for patients. And that's what we think makes Tenaya different. We have this commitment to not only the discovery, but the development of treatments of genetic heart conditions. And our science uses genetics to identify new drug targets for rare and prevalent forms of heart disease. Our current pipeline, which you'll see in a minute, includes 5 programs being developed for over 7 different heart conditions. And I think we want to spend most of our time this afternoon and this evening is really exposing you to our gene therapies and the opportunity to deliver a working copy of a gene to potentially restore the function or prevent the progression of heart disease. So why do we think that the time is now, and why do we think genetic therapies are emerging and going to be at the forefront? Well, for the first thing, heart disease is not going away. Heart disease is still the leading cause of death worldwide, so the need is still great. You combine that with increasing genetic insights and new ways to diagnose diseases and cardiomyopathies, we really feel that that leads us to where we are today, and that is combining the expertise in cardiovascular and genetic medicines to treat the underlying causes of heart disease. So not just treating symptoms, but understanding what is fundamentally the issue that drives the disease process, and then go after that to target this underlying cause of heart disease. This is reflected in our pipeline and in our programs and the indications, where I won't go through each of these, but to say that we'll spend a little more time, of course, on our gene therapy programs, which are the TN201 and particularly the TN401, the gene therapy for PKP2 associated ARVC. So let's spend just a minute in some of the basic terminology, the basic understanding of genetics, and then it leads us to a better understanding of what gene therapy is and what it may do. So of course, at the basis of all of this is a cell, and a cell, of course, with a nucleus. Inside of the nucleus are 23 pairs of chromosomes. Chromosomes consist of strands of DNA, which are the basic codes for the building blocks, which are proteins that are the building blocks for all of our organs, the various structures within the cells, and are, again, kind of the Lego building blocks of the body. So each of these strands of chromosomes, strands of DNA, contain discrete genes, and these genes then encode very specific proteins. So there are over 20,000 genes contained in all of these chromosomes. So it's just a marvelous wonder how this all ends up coming together. But we know that it can be disrupted. And we know that changes in genes can lead to genetic conditions. And many of you that are hearing this evening are probably affected by one of those gene mutations. So what is a mutation? It's a change in the DNA coding, and it can affect then the gene's ability to make fully functional proteins. And by just altering the code in just a small portion of the DNA, you can lead to changes in the proteins that actually make them dysfunctional and can lead to various heart conditions. So these mutations, also known as variations or changes, do cause genetic conditions, but some do not. And many heart conditions have an underlying genetic cause, and certainly ARVC is one in which a number of genes have been identified. And these gene mutations can be passed down from parents to children, or inherited, as we would say, but they can also be caused by environmental exposure. So hopefully that helps you understand what a mutation is and how it can affect function of the heart by the alteration in the code, and then ultimately the protein, which is the building block. So why then the promise of gene therapy? And what is it? It's certainly an investigational approach to treat genetic heart conditions. It's not necessarily completely new. There are actually 2 approved products based upon a gene therapy approach. One is for spinal muscular atrophy, a neuromuscular condition predominantly affecting children. And the other is to treat various forms of retinal disease. So this is not completely new. There are a couple that, as I say, are FDA approved and being administered to patients today. However, not for heart conditions. And Tanaya, as well as others, hope to be the first to be able to bring this forward to treat various forms of medical disorders. So the basic concept is that we introduce a functional copy of a gene into the cells so they can carry out their typical activities. As I shared with you in the prior slides, the fundamental cause of the disease is a mutation in the coding. What we would do then is introduce a gene that has the normal coding, so that can then lead to copies of the normal and functional protein and restore kind of the normal building blocks of the structures involved. What's so unique about gene therapy, it has the potential to correct the underlying cause of genetic heart conditions. And we think that's what's so exciting and the great potential here in gene therapies. I think what is important to note is it does not make changes to the genes in a person's sperm or egg cells. So it doesn't fundamentally alter the genetics of an individual such that the working gene cannot be passed from a parent to a child, and receiving gene therapy will not change the risk of passing on a genetic condition to children. And I think that's a very important point to make, particularly about the gene therapy approach that Tanaya and others are embarking upon. So what do we see as the goals of gene therapy for genetic heart conditions? Again, just to re-emphasize that we can truly target the underlying genetic cause or root cause of these conditions. And that would allow us then to potentially prevent or actually reverse the underlying condition. We envision this as being administered as a single one-time intravenous dose that will have long-lasting effects and confer a long-lasting response. And ultimately, this would demonstrate meaningful improvements in heart rhythm, heart pump function, symptoms, and I think very importantly, the quality of a patient's life. So this is really the interesting and fun part. How do we do this? We talked about what genes do, what a mutation does, and what we hope to achieve by introducing a new gene as part of a therapeutic approach. But how do we do that? Well, we use a virus. It's called an adeno-associated virus, or AAV for short. Certainly through these past 2 years, we probably have all learned more about viruses than we care to know. But believe it or not, there are some good viruses, and we've also figured out how to harness some of the things that a virus can do and harness that and turn that into something very useful and effective. So these AAVs are modified for use as a vector or a delivery vehicle for the new gene. So these vectors have the ability to deliver a working gene to target cells. These the various genes of the virus are removed from the vector, so the native genes intrinsic to the virus are removed, and then it's replaced with a working copy of the gene for delivery into target cells. So the advantages of AAVs as a gene therapy vector include the fact that they're very efficient at getting into cells. They don't cause any diseases in individuals. They occur naturally in the environment, and various types of AAVs exist and can be tailored to target specific types of cells. And certainly in the case of Tanaya and others looking at the heart cells as being the target, there is one of these AAVs that has tropism or a particular affinity for heart cells, and we've been able to harness that as part of our approach to make sure that the virus can predominantly go to heart cells as opposed to some of the other cells and other tissues in the body. So these vectors contain the working gene, and there is contain what we're terming an expression cassette, which is the gene therapy package. So this it contains what we call transgene, or is the new functional copy of the gene, is the transgene. Its on-off is directed by a promoter that is specific to the cells, and in our case, we're using promoters that are specifically turned on in heart cells and not effectively turned on in others. And there are other regulatory elements that are needed for this working gene to be converted into a functional protein. So again, we've removed what is normally within the virus. We've repackaged a new working copy of the gene, which then can create that new functional protein, which is the essence of the process of the disease. So to kind of put this all together in one diagram, that the viral DNA is removed, the working copy of the gene is inserted into this viral capsid. The treatment is administered by a one-time intravenous infusion. As I noted, it can target muscle cells in the heart due to this specific type of an AAV, which does preferentially go to heart cells. This vector then delivers a working copy of the gene to the heart muscles. Then the working copy of the gene restores the function to heart muscle cells to treat this genetic condition. So there are risks, and I think the field is learning more and more about these risks, how to anticipate them, and how to treat them. But nonetheless, we'll review them here. The immune system may mistake the gene therapy as a potentially harmful intruder and can trigger an adverse immune response. If this happens, the treatment might not work as it is intended. In studies of AAV and other forms of gene therapy, the researchers have observed liver inflammation in some people. Currently, a person can receive an AAV gene therapy only one time. It cannot be redosed or administered in a larger dose at a later time. And that's really because a person's immune system will have made pre-existing antibodies to that vector that is used in the gene therapy, and those pre-existing antibodies will attack subsequent doses of the treatment before it has a chance to work. We know that AAV gene therapy may deliver a working gene to the incorrect cell as well. Participation in an AAV gene therapy clinical trial may prevent an individual from participating in clinical trials for other AAV gene therapies in the future, mostly because of the cross-reactivity of the immune system to various types of these vectors. And please note as well that AAV gene therapies are still under investigation, and not all of the risks are known, although we are actively investigating, we and others are actively investigating. But I think to end really with what are the potential benefits of gene therapy for individuals with ARVC. So while gene therapy for this inherited heart disease is still under investigation, it has several potential benefits. Again, the benefit of being a one-time administration, a one-time therapy, to actually stop or reverse the ARVC progression, to stop or decrease the need for other medications and devices, and to ultimately increase the quality of life. So let's spend just a moment talking about the TN-401 gene therapy for individuals with ARVC with a PKP2 gene mutation. So I can't teach you about ARVC. You can probably teach me. And I'm sure most of you know that the most common cause of ARVC is a mutation in the PKP2 gene. If you look at the rough numbers, there are about 70,000 individuals with a PKP2 mutation in the US alone. And we know that just one copy of the mutation from a parent to a child can cause ARVC. So this is then termed as an autosomal dominant, because it only takes one copy of the mutated gene in order to potentially cause ARVC. So to go back to some of that we set up earlier, in that a mutation in the coding of the gene for PKP2 can lead to a dysfunctional protein. In general, that means that there is not enough, certainly not enough of the normal protein that is made, and it cannot form the structures that it needs to form in the heart cells, and this ultimately results in ARVC. So again, it starts with that fundamental change in the code that leads to altered protein. And that's why, again, the promise of gene therapy is to correct that and to normalize the production of the protein. So we are developing a gene therapy for ARVC that is caused by these changes in PKP2. It's called TN-401. We're targeting ultimately cardiomyocytes, or the cells in the heart that make it contract, and also the electrical conduction of the heart. So the goal is to add a working copy of this gene to cardiomyocytes to restore the expected protein levels. Then that would allow these cardiomyocytes to function in the usual way. We are starting now, and this is kind of a prelude to the interventional study. We're initiating what we call a phase 0 study to understand the amount of intrinsic AAV antibodies in people with ARVC who are also PKP2 mutation positive. As I shared with you earlier when we talked about the AAV virus, these are naturally occurring. You could be exposed to this without knowing. It does not cause disease, and yet you would mount an immune response to having been exposed to the AAV virus. So the concern would be that if you have too high of a level of an immune response that is already programmed, that the therapy might be less effective or ineffective, because the immune system would attack the very vector that we need in order to deliver to the cardiac cells. So that's part of the understanding that we want to have, is to study and identify patients then with ARVC that are caused by a change in the PKP2 gene that then would most likely be eligible and benefit from Tenaya's investigational gene therapy program. And part of that is characterizing your prior or existing immune response to the AAV virus. So this study that we will be launching by the end of this year, participants will take place in the study for 5 years. So you'll be followed as your usual care, usually annually. And these annual visits will include a blood draw per standard management and with your participants, physician and specialist. So you would go to the caregiver that you would normally visit as part of this program. The goal is to enroll about 200 patients. This ultimately will be a global program, not only sites in the United States, but sites over in Europe and perhaps Australia as well. So the study, we hope to initiate the study by the end of this year. So why are we excited about the potential for this TN-401 for the human disease? Well, it's because of the preclinical data that we have in a mouse model of ARVC and the effectiveness that we've seen of a copy of the mouse gene. So it's the same approach to actually decrease the function of the gene in the mouse and then to restore it by introducing through the adeno-associated virus a new copy of the gene, the expression of normal levels of protein. And what we see is that we're able to prevent the development of the disease in mice, that indeed we can induce ARVC by removing the PKP2 gene in the heart cells. So if you follow from left to right and you look at the color coding, the black line is normal healthy mice. The blue line is mice lacking the PKP2 gene, and the red is the mice lacking PKP2 gene but treated with gene therapy. And as you can see on that first leftward panel, and we're looking at the heart enlargement that occurs over time in this mouse model, the treatment with the gene therapy in the red brought it back to near normal as compared to the healthy mice, whereas those that lacked the PKP2 gene and did not receive gene therapy, their heart enlarged, as you can see in that blue line. In the center panel, not only did the gene therapy prevent heart enlargement, but it prevented the loss of heart function or the worsening of heart function that occurred in the mice population that were treated only with the placebo. If you look there in the blue, it is a much lower ejection fraction or measure of heart function as compared to the healthy mice, whereas those that were treated with the gene therapy have essentially normal heart function. This actually translates into a survival benefit. And if you look at this curve, each downward tick is the loss of one of the mice. And you can see the blue line as those that were lacking the PKP2 gene that were not treated. Essentially, all of the mice had died by 6 to 7 weeks after the initiation of the disease model. In contrast, the mice treated with the PKP2 gene therapy in the red line remained healthy and alive throughout to the end of the study. So again, quite a very promising and quite dramatic effect of the gene therapy in this rather severe disease model in mice. So not only did it have the effects that you saw in the prior slide, but as you know, particularly those that are affected by PKP2 gene mutations, it tends to be very arrhythmogenic. So one of the key questions that we wanted to address was whether or not treatment with gene therapy could indeed prevent the abnormal heart rhythms that are part of the ARVC model in mice and also seen in patients. So you can see, as we're calling this a normal rhythm strip, you see these spikes on this strip as being roughly the same in size and at a very consistent cadence. If you look first at the bottom, the PKP2 mice that were treated with the placebo and not the gene therapy, they show a lot of abnormalities on the heart rhythm strip, non-sustained ventricular tachycardia, premature ventricular contractions or PVCs. And these are characteristics of some of the things that are seen in patients in the clinic. However, in the middle panel, the mice lacking PKP2 but treated with the gene therapy maintained that normal and usual heart rhythm. So this was very encouraging that indeed we could address the underlying rhythm abnormalities that are seen in patients that are affected by PKP2 gene mutations. So where are we and what are our next steps? Certainly this is part of our outreach to the community. We want to learn from you just as we want you to learn from us. And we've been very active in developing education programs, and we want to learn from patients. We've had individual meetings with patients. We've had roundtables with patients affected by ARVC. And again, our mantra of patients first, we translate this into wanting to hear from you along the entire research and development continuum. So this is part of what we're doing. Perhaps those of you listening to this have participated, and certainly we want to thank you for doing so, and we look forward to your continued support and participation in these types of programs. And we want the community to be well informed, to be able to have the opportunity to hear about these novel programs, to be able to share with us what your hopes, what your concerns, what your questions are as you think about whether or not a therapy like this down the road would be appropriate for you, and to make sure that you begin to have those conversations with your care provider. As I shared with you, we're very much active in bringing this forward to the clinic. We have been speaking with probably many of the physicians who have been caring for you. We expect to have this phase 0 registry and study, as I shared with you, operational by the end of this year. And then we actually expect to file an IND or investigational new drug application as early as next year. So really the bringing this to the clinic is literally just right around the corner. Another key component that we didn't spend a lot of time on talking about, and that is manufacturing. We have made the commitment at Tenaya to be in control of the entire manufacturing process to assure the quality, safety, and ultimately the efficacy of the gene therapy that we bring forward. So to summarize, Tanaya Therapeutics is committed to understanding the experience of patients and to make sure that we incorporate those learnings into our drug discovery and development efforts. And in order for us to create more effective treatments, we identify the genetic causes that are disrupting the healthy heart function, centered on gene therapy, because we know that gene therapy is a potential treatment for heart conditions such as ARVC. We have designed to provide a working copy of a gene to the body to create a new functioning protein. We've tried to share with you some of the risks and benefits of gene therapy that do exist, and individuals that are considering participation in the clinical trial, we certainly encourage you to begin to have that conversation with your physician. We are developing the TN-401, that is a gene therapy for individuals with mutations in the PKP2 gene. Down the road, we will be looking at other causes and other genes that are mutated that cause other forms of ARVC, as well as other forms of heart disease. So with that, I want to make sure that you're introduced to our patient advocacy, Lori Correa, who is on this call. This is some of the contact information in case you would like to reach out to them. And if you have any questions about the TN-401 program, or if you're interested in participating in our roundtable or in Tenaya's Patient Advisory Council. So we welcome your active participation, look forward to it. We always appreciate it, and we always learn from it. So with that, I will close and make sure that we have enough opportunity and time for your questions and very much look forward to the interactions. So thank you so much for your attention. And again, I look forward to your questions.
Thank you very much, Dr. Pollman. One question here, wondering if gene therapy will last for my whole life.
Great question. That is the hope. What are we building that hope on? Well, you got to start small and then work large. And certainly in the mouse models in which we induced disease that is very reminiscent of ARVC in humans caused by a disruption in the PKP2 gene, we saw that the effect lasted for the lifetime of the mice. We've also seen this in another gene therapy program for a different indication, where again, the effect lasted the entire lifetime of the mice and was able to maintain the normal heart function and normal heart structure throughout that lifetime. We also know that there's emerging data from some other programs of gene therapy for heart disease, where they're seeing sustained expression of the new gene and the new protein that is made. So I think all indications are that this could be a long-lasting therapy, and it would be our hope that this could truly be kind of, as we term, a one and done and a lifetime therapeutic.
Thank you very much. Let's see, from Mohit of Regeneron, does gene therapy is more of a preventive or a rescuing? Can it reverse cardiac disease progression associated with monogenic mutations in heart?
Yeah, great question. I think we don't know the complete answers. I think there is hope that we can reverse many of the processes that have already taken place in many of the disease states. Certainly in some of the animal models, we have seen evidence that we're able to reverse many of the processes, adverse processes, once they've been established in the disease models. I think the model that I shared with you as it relates to ARVC was more of a preventative model, because the disease progresses so rapidly in this model. It's just in a matter of just a couple of weeks. Once we alter the normal PKP2 expression in this particular model, the disease rapidly progresses, and it's almost impossible to reverse that particular model. So we're looking at other models in which we could better address that question of how much is reversible versus preventable. But our hope is much of this is actually reversible. I'm sure there are some elements that aren't. As you may be aware, there can be a fibrosis and fibrofatty replacement process as part of ARVC. It is unlikely that we would be able to reverse that process once it's established. However, we certainly could prevent the further establishment and progression of fibrosis and the fibrofatty. It is our hope and belief, based upon experimental data, that the process such as the arrhythmias that cause the arrhythmias, that we would hope that we could reverse that. But again, I think we need better models to demonstrate that. So a very good question, and appreciate that.
Okay, thank you. Another follow-up question from Alice was, can you have gene therapy again? Can it be repeated?
Yeah, so the challenge that the field faces now is the fact that once you are administered an adeno-associated virus, that your body does mount an immune response. So you will have a memory of being exposed to that, and that memory can be activated fairly quickly if you're re-exposed to that particular virus. So the concern is that if you're re-exposed and you have this response, that the therapy might be less effective, and you might have a fairly robust immune response that might adversely affect the patient who receives an additional dose. So as it stands today, the answer would be no. You can only receive this type of a therapy one time. Is that going to stay that way? We hope not. And there are certainly active programs, both in academics as well as in companies, to help us understand what drives that process. Are there ways to blunt that process and maybe even prevent that process, such that an individual could receive a second therapy if needed? So hopefully that addresses where we are, and also the activities around where we hope to be in the future.
Yes, thank you. Excellent response. Any more questions from our community? You can post it in the chat, or you can even unmute yourself and ask it verbally. I don't see any more questions at the moment, Matt.
Okay.
So I want to thank you for your fabulous presentation and addressing the few questions we had. And so I thank Tanaya Therapeutics for their support to the SADS Foundation. Oh, there's a few more questions are coming in now. So let's
Super.
go back to
Yeah,
this. All right.
love the questions.
Marco asks, how young can a person be to receive this gene therapy?
Great question. You know, some of the gene therapy that is actually being administered today for other indications is in very young. Infants, children affected very early in their lives by neuromuscular disorders. So there's no reason to believe that ultimately any of these gene therapies could be delivered to very young patients. One of our programs is anticipating treating newborn infants who are severely affected because both copies of a particular gene have been mutated. And without a transplant within the few months of life, they actually would die. So it is very possible, and there's precedent out there right now to treat very young, as well as older patients. So I think there's the entire spectrum of age. It really all depends on the severity of the symptoms of the disease as to what and when is the most appropriate time to treat any individual patient.
Okay, thank you. So Christy asks if you're starting any research on other genes. She's putting her vote in for DSP.
Excellent. I can say certainly yes. And we recognize that in particular DSP, there is a more of a predilection to heart failure earlier and more severe, certainly than is normally seen with PKP2. So we recognize that the need is great. I think to get into some of the technical reasons why it's a little bit challenging, just to build upon our learnings and understandings of that virus, the AAV itself is limited as to how much of a coding sequence you can package inside that little capsid or that delivery envelope. And DSP is a little too large to be able to conveniently and effectively package into the delivery systems that we have today. Does that mean that we won't get there? I sure hope not. And we're constantly looking for new ways to package and to be able to work with larger genes, because many of the genes, other genes that cause ARVC have this same issue, that they're a little bit too large to package today into the AAV virus. But we're working on it, as are others, and I'm very hopeful that we'll get there.
Okay, thank you. Mohit asks, how fast does it work after the dose?
Yeah, wow, another great question. If we use the animal models as a guide, it's just a matter of a few weeks before we see fairly robust expression of the introduced gene and protein. So it can be fairly quick in terms of when you start seeing the new gene and protein expression. Depending upon what you're trying to target, it may take a little while to kind of normalize and equilibrate as that new protein needs to figure out how to effectively integrate into the structure that it has been disrupted. But it's not months; it's really weeks when you start seeing that expression.
All right, thank you. Darcy asks, do we have to be in a certain location to receive gene therapy or receive it from our hometown cardiologist?
Yeah, as we envision kind of the early stages of clinical trials, they will be what we would term as centers of excellence who have had experience with cell or gene therapy in the past. We would hope to set these up in kind of geographical locations that would be fairly convenient for most patients who would want to participate to travel to. But it's unlikely that in a smaller city, smaller town, that one of these centers would exist and be able to treat patients. We want to make sure, particularly in the early phases, that that whoever receives this type of therapy is under the best care possible, is monitored as closely as can be, and we can see hopeful signals as early as possible. So these would occur in geographically located centers of excellence.
Thank you very much. Okay, Hisham says, can you please expand on the TN-201? Where are you in the process? I guess this is a more advanced platform in your pipeline. What are you seeing in the initial experiments? Yes. The HCM community is waiting eagerly with great hope.
Great. No, thank you for that. Yes, our TN-201, of course, we didn't focus on that in this presentation, but essentially, as we have made public statements around the 201, we expect actually to file the IND for the FDA approval to begin to initiate trials. So to the filing of the IND to occur this year. I think we've been able to share some very encouraging and promising data as it relates to mouse models of the disease caused by a lack of the MYBPC3 gene and protein. And in that model, again, we've been able to see fairly dramatic effects in reversing some of the early indications of disease with reduced ejection fraction, enlargement, thickening of the heart. And those have had sustained effects all the way through the natural lifespan of the mice. So very encouraging preclinical data, filing an IND this year for that program. So again, very hopeful that we'll be able to start bringing this to the clinic soon.
Thank you very much. Are there any other questions? Okay, it was a fabulous presentation, Matt. Thank you very much for your time and your support for this. So I wish everyone a good evening and thank you for your participation and joining us tonight.
Thank you, Marcia. Thank you to the participants as well.